Hi, so this is the first of 3 videos for this week, and we're moving into our next topic, which is topic 15. And the start of this topic is all about variation within populations, and it draws on the topic knowledge of topic 14, which is the mutation stuff that we covered last week, but also your knowledge of meiosis and how meiosis produces variation within populations. And then this week we're going to introduce a little bit more of the statistics. So I know that we've already had a go at standard deviation and understanding what standard deviation means within data sets and using that when comparing population data. We'll be doing more of that in today's video, and then in the next video we'll be moving on to our first of 3 stats tests that you need to know, which is the t-test. And then finally, towards the end of the week, we are going to be doing natural selection, which is very similar to what you studied at GCSE, a little bit more detailed and some more key terms, much more application. So that's some fun to be looking forward to. So let's get started. Okay, so our starter is a review of your GCSE genetics. So I'd like you to pause the video and have a go at this genetics problem, which is from your GCSEs. Okay, so hopefully you've had a look at this question. So in this question, you've got a type 1 tomato, which you'll remember being described as homozygous dominant because it has 2 dominant alleles. Okay, and a type 2 tomato, which is homozygous recessive. So being asked why, if those 2 tomatoes were bred together, that all the offspring would have the characteristic of being red. So we're looking for you to remember how to do your Punnett square, your genetic cross. So understanding that for our homozygous dominant tomato, when it produces its gametes during meiosis, because it only has this dominant allele present on both homologous chromosomes, the only variant of gamete that it can produce is one with the dominant allele. And equally for the homozygous recessive individuals, that type 2 tomato in the question, it only had 2 copies of the same recessive allele. So the only type of gamete that it can produce is one containing the recessive allele, which means that all of the offspring would be heterozygous. Just correcting a typo. So all heterozygous, which means now all the offspring will carry one dominant allele and one recessive allele. But because the dominant allele is a dominant allele, it will be the allele that is expressed, even if it's only present, if it's present with another allele, okay, because it's the dominant one. So all heterozygous, all the offspring are heterozygous, so they all have one dominant R allele, and you only need one copy of your dominant alleles for them to be expressed in the phenotype. So all the offspring will be red. Now, if you enjoyed this part of your genetics in your GCSE studies, you'll be happy to know we're going to be doing lots and lots of genetics in year 2. Okay, and it gets even more complicated than this by then. But the reason I've put this in at the start of this topic is really so that you can link your genetics from GCSE to what we've learned about in meiosis at the moment. So when we're talking about alleles, you are representing them with large letters, capital letters for dominant alleles, and small letters for recessive alleles. And so you were talking about alleles, you were talking about organisms having certain characteristics, like being a red tomato, because of the alleles that were inherited and the combination of alleles inherited from the parent plants. So you were already thinking about this at GCSE. So we're taking this a little step further in this topic today. So you're working in your topic 15 student notes, Okay, and we're actually working alongside the notes, so you can have those open and use those, and I'll bob into them now and again. So I'd like you to have a go at answering these questions. Now you can do it either just from what you think, from your background knowledge, or you can have a little read through the beginning of the notes and use that to come up with your answers. But if you pause now and give these a go, and then I'm going to go through the answers for you. Okay, so the first question, what is variation? Okay, so at the beginning of your keynotes, you'll see a definition. All right, so what is variation? Well, variation is the differences between individuals. Okay, so within one population, like our little litter of kittens here, we've got definite variation in the patternation and the colour of their fur and the eye colour, but obviously other things too. Okay, so these differences, that's what we're talking about when we talk about variation. So what causes variation? Oh, sorry, what types of variation are there? I forgot I popped this in. In your keynotes, you'll find that you've got 2 new key terms to describe different types of variation. Most of the time when we're talking about variation, we're talking about variation within a population of the same species. Okay, this is called intraspecific variation. So sometimes, and in our next topic, we'll be talking about variation between 2 different species, and that is called interspecific variation. Okay, so what causes these types of variation? Well, if we think back to our kittens, why have we got this variety of patterns in fur colour and patternation and eye colour and length of ears, etc., presence of a tail or not, all those things? What's causing that variation within that population? Okay, so you should have some ideas forming about what causes this kind of variation. Normally in class we kind of stop here and have a go at thinking about the variation that we have in our own population, our human population, just within that small group of people in the classroom. So I thought it'd be interesting for you to have a go at home. So you could now, for the individuals of your family, find out what your blood groups are, what your hair colour is, whether anyone suffers from red-green colour blindness, and the way you would assess that is if they have a look at this circle here. If you can't see the number 27, then that would indicate that you can't see the difference between the green and the red spots, and so are red-green colourblind. Though they will probably know. And their height. Okay. Now, the idea of exploring these variants within our population is all the time so that we're aware that there's variation within our population, but also that some types of variation have more causes than others. Okay, so if you've thought already about what causes variation, now within that table, as you're writing that down, are any of these caused by a wider variety of factors and others are fewer? Okay, so what causes the variation? Well, all variation within populations is the result of 2 things. Well, one of 2 things. Genetic diversity, now that's always a reason for biological variation within a population, but for some characteristics, there's also an environmental influence. So let's pop back to the ones that you're investigating at home. Which of these are just the result of your genetics, so genetic diversity within the population, and which are influenced by the environment? So let's take the first one. What do you know about blood groups? Okay, well, blood groups in human population, there are only 4 different types. Now I'm ignoring rhesus positive or negative here. So people are either blood group A, B, AB or O. Okay, so everyone in your family will fall into one of those categories. Okay, so what do you think controls that type of variation within a population? Well, it is completely genetic. Okay, so there is one gene and there are actually 3 alleles for that gene, and they display a pattern of inheritance called codominance that we're going to look at next year. So there are only 4 varieties of that characteristic in the human population, and it's down to the expression of one gene. Okay, thinking about her colour then, well, we know that her colour is quite a lot more complicated. So if we just consider natural hair colour at the beginning, we know we've got quite a number of different variants in the human population. So if it's genetic, it would be controlled by more than one gene, influenced by more than one gene. Okay, so if you think back to our pussycats, okay, they don't visit the salon, but they've already got a great variety in fur colours, and that's genetically controlled in these cats. Okay, but not by one gene. There's often more than one gene, and they're interacting together to give you this big variety. Okay, so some are controlled by one gene, some are controlled by many genes. Okay, for humans, of course, we've got the environmental influence of hair colour as well, because some of our hair colours is not natural, so we also have the environmental factors. Red-green colour blindness, okay, that is controlled by one gene, and so it is completely genetic in that variation, and there is only a variant of 2 to be colourblind or not to be colourblind. And then finally, if you think about height, this one is very much a mixture of both. So there are a number of genes that influence our height, but at the same time, how much food we have available to eat during our childhood and later on would also affect whether or not we reach our genetic height, if you like. Okay, so the human population in terms of height is a massive range of different heights, and we can call this kind of variation continuous, because there is basically someone of every single height, rather than falling into categories like small, middle and large. We actually have humans of every height across the whole range, from the very shortest to the very tallest. Of course, most of us fall in the mean of that, in the middle. Okay, something that will show you a little bit more about how much influence the environment has on our height is if you consider what's happened to height over the last century. So this is the average height of a male aged 21 born in different time periods, and you can see that there is a very strong trend for an increase in male height over the last century. Now, when you think about genetic diversity, it's possible for a population to change in its genetic diversity because of natural selection, but that process takes a very long period of time, far more than a century. So scientists believe that our increased height over the last century is not really to do with changes in the alleles within our population, our genetics, but much more to do with the availability of food and children no longer having to work, etc. Okay, so you can see that that shows a really strong influence of the environment for this particular characteristic. So variation are the differences in individuals within a population, and it's always caused by genetic diversity within that population, and also for some characteristics, genetic diversity and the influence of the environment. So anything that shows this kind of continuous variation is more likely to be influenced by the environment, like our height. So when you're looking at data sets, if it shows this kind of pattern of a normal distribution curve, it's either going to be controlled by genetic diversity, but many different genes, but more likely to be an interaction between genetic diversity and the environment for that particular characteristic. Okay, so I also asked you about this genetic diversity, so the cause of this variation. Okay, sorry, no I didn't. I asked you what the cause was. So we're going to explore this a little bit further. Okay, so what's the cause of the variation? So we're going to have a look at the genetic diversity, because there's quite a lot more to talk about in there. How does genetic diversity produce variation? Okay, so genetic diversity is a measurement of the number of different alleles of genes in a population. Okay, so if we consider this population of beetles, okay, within this population there are 3 varieties of beetle colour. And the reason there are 3 varieties of beetle colour are because there are 2 alleles, and for this particular characteristic, if you're homozygous dominant, you have dark coloration. If you're homozygous recessive, you've got a light coloration. And if you have a heterozygous, you have 1 of each allele, because there is no dominance in this particular characteristic, you get a colour in between, so a brown. Okay, so there's 3 different varieties controlled by 1 gene, of which there are 2 alleles. Okay, so that's what genetic diversity is. What kind of factors affect it? Well, one incredibly important factor that can increase genetic diversity is mutation, because mutation is how we produce new alleles. So have a think about this mutation here. Let's do a little bit of a recap on our types of mutation. Can you stop the video and have a go at describing what mutation has occurred? So this is the original mRNA and this is the mutated mRNA, and describe the impact of the mutation. Can you also describe what's happened to the DNA in that mutation? Okay, if you're ready to go through, I'm going to go through the answer. So the mutation was the third codon, okay, and it was the first base. And if you have a look, it was a substitution. And when this substitution occurred, remember it would have happened in the DNA template during DNA replication, and it means the third triplet in this code changed from GGG to TGG. Okay, so remember this would be the complementary triplet to that codon, yeah, because this is the template strand. So describing this mutation, it would have been a substitution, so we're swapping one base for another, and it was guanine to thymine in the DNA, which meant that in our mRNA we've now got cytosine, we've replaced cytosine with adenine. The impact was we changed 1 amino acid in the primary structure of the polypeptide chain. Okay, so we've got a new allele. Okay, so this new allele could be a beneficial allele to the organism. It could be a better protein. Okay, I know in the last topic we just concentrated on that mutation having caused a change to the primary structure, that it would mean that the protein wouldn't work very well, wouldn't function, would be a different shape. But what if that different shaped protein was actually a better shape, or did something different and gave the organism a different characteristic? So that's what we've got to start thinking about in terms of mutation. No, it's a process that produces new alleles, and sometimes those new alleles don't cause problems, they cause variation in the organisms. Okay, so have I got another mutation? Can you describe the type of mutation, what's changed in the DNA, and what impact it's had on the primary structure of the protein? Okay, so if you're ready to go through, I'm going to go through the answer. So this time it was a second codon. Here we've actually got a 2 base deletion. Did you spot that? And that means we've got a frame shift, because we've got a deletion and addition mutations always cause a frame shift. And in this case, we had lost from the DNA the bases that coded for the U and the A in the mRNA. So we lost the adenine and the thymine at that position in the DNA molecule. And that has meant a shift along to the left by 2 bases, and the impact has been felt by downstream of the mutation by every single codon. So each codon has changed, so each triplet in the DNA changed, and each codon in the mRNA has changed, so we've now coding for a different polypeptide chain. Okay, so again, this kind of mutation could result in a new allele, a variant, a new characteristic in the population that could be of benefit to the population. Okay, so mutation then, we've now got to start thinking about in 2 ways. So sometimes mutations cause faulty proteins to be produced and result in a genetic disease like cystic fibrosis. But equally, sometimes mutations produce a new variant, a new allele, and so a new variant of a particular characteristic in the population. So for example, imagine if the gene for beetle colour mutated and we got a new allele, okay, that when that was expressed by the beetle resulted in a different colour, a new colour in that population. Okay, so mutations are very important because they are the only way we can generate new alleles in a population, which give us great variety in that population. So some other things that we know produce variation, okay, although they won't produce new alleles, during meiosis we mix alleles, okay, by 2 processes that we know about already: independent segregation of homologous chromosomes. That is one way we mix the alleles that we've inherited from our parents around to give new combinations of those alleles to our offspring, and also via crossing over. Okay, so again, during meiosis, the swapping over of the ends of non-sister chromatids within the homologous chromosome pair means that we are mixing alleles that we inherited from our parents, so that when we pass them on, we're actually passing them on in lots of different varieties. So if you have a look at this example here, this would be the chromosome inherited from mum, and this from dad. Okay, but because of crossing over, when this organism passes these alleles on to their offspring, they're actually passing them on in the form that they inherited them, but also in a new mixture. So you can see here, one gamete is actually going to contain the dominant A and the dominant B, and one the recessive A and the recessive B, which of course they didn't inherit that combination. That combination has been produced during crossing over in meiosis. So both independent segregation and crossing over mean we are mixing the alleles as much as possible that we inherited from our parents during meiosis to give us new combinations that we pass on to our offspring. Okay, so then the final mixing, if you like, is via fertilisation. And this is what you were showing in your GCSEs all the time, with this idea when you were doing your Punnett crosses, that when egg and sperm fuse, or when gametes fuse, we bring together alleles from individuals, okay, but only one allele from each individual. So to mix them again, okay, but of course it's a random process. Thinking about the sperm penetrating the egg, which sperm actually penetrates the egg is random, isn't it? Okay, so which of these combinations, we've got an equal amount, an equal chance of them occurring. Obviously for our heterozygous combination, that's more likely from these two heterozygous individuals. So we can work out ratios, and I'm sure at GCSE you talked about there being a 1 in 4 chance of two heterozygous individuals producing a homozygous dominant offspring, 1 in 4 homozygous recessive offspring, and a half, yeah, 50% chance of a heterozygous offspring. Okay, so there's an equal chance of all of those fusing, and that's because this process is random. So now, if you think about the causes of variation, they are genetic diversity, and into this we factor mutation, the mixing of alleles during meiosis, and random fertilization, and the environment. Okay, so all of these things make for a great variation in the population. So why go to all this trouble? Why is this variation so important? Why do we need new alleles? Why do we need to keep mixing alleles? Well, if you think about variation and where you've met that key term before, it was at the beginning of your natural selection story at GCSE. So you always started with, there is variation in the population. Without variation, natural selection could not happen. Okay, so variation increases the chances of a species surviving via natural selection. It increases the chances of offspring having characteristics that will help them survive in new conditions. So if conditions change and we've got a big variety of different characteristics, we'll be much more likely to have characteristics that are favourable to that new environment. And that is, of course, what natural selection is all about. You can't have selection unless there is a variety to select from. Okay, so of all of the things that cause variation, mutation is the most important one to natural selection, because it's the way we generate these new alleles. So if you imagine this beetle population now with our new allele generated, imagine that suddenly the population finds itself in an environment that is very much more green. And having this new variant, this new allele, provides you with better camouflage. We would expect over time that the individuals that have this new allele will be more likely to survive in the green environment because they'll be less likely to be eaten by predators. And so it will be the advantageous allele to that population. And over time, it would increase in number. So we'd see more of the individuals in the population having that allele present. But that would only be present because we've got our new allele. So if we didn't have our new allele and we play the story out again, it becomes very green, okay? Then no individual's got an advantageous allele, and we might result in the beetle population becoming extinct. Okay, so a couple of points to ponder. So particularly when we think of the human population and our characteristics, many of them are both our genetics, so what the genes we inherit, our genetic diversity, the alleles and their combinations, and the influence of the environment. So what kind of people do you think might be useful in scientific studies to determine if a characteristic is genetic or environmental? So a good example here might be IQ or intelligence. So there have been many studies done to assess how much of that is controlled by genetics and how much by the environment. So what kind of people do you think might be useful for that? So that's a point to ponder for a second. And then when we're studying variation, we can't possibly measure the variation of the entire population. So for example, with the beetles, I can't record the colour of every beetle in the population. Okay, but what I could do is take a sample of the beetle population and then count how many of each colour are in that sample, and then apply that to the estimate for the population. But when I do that, how do I make sure that the sample that I take and then the estimates I make are valid? Okay, so that when I compare groups of individuals in different populations, how can I make sure my conclusions are valid? Okay, so back to the top one, what type of people would be useful to determine if a characteristic is genetic or environmental? Identical twins. Okay, so identical twins are genetically identical. Therefore, any difference in a characteristic that is measured between them, we can say is due to the environment. Okay, so we can make measurements like that. And there have been studies like that, and there is a question in the question pack that's based on that idea. Okay, so measuring variation. So this is in your student notes as well. Okay, so measuring variation, we can't measure every individual in a population, so we need to be able to take a sample. When you take a sample, you need to design your study so the sample you take is random. Yeah, this is to remove bias, and is large enough to be representative of the population. So you need to take enough samples that your sample results will be representative of the whole population. Okay, now when you collect your data, you calculate a mean value for your collected data, but you also calculate a standard deviation. Okay, now you don't have to learn how to manually calculate a standard deviation of a mean. Okay, you can use Excel, and in Excel you can simply type in equals standard deviation and highlight the data, the full set of data for that you want the standard deviation for. Okay, so that's really simple, and it's something that we need to do in our practicals, the ones that are coming up, but it's not something you will ever be asked to do in an exam. Okay, but standard deviation is really useful, and the examiner uses standard deviation a lot to allow you to interpret mean values and compare them to conclude if any differences you see are significant. Okay, so we've done some work like that before, and we're going to continue to do that in our variation topic. Okay, so when we go back to college, hopefully we will have some time for some more investigating variations in our groups. Okay, so we will do a number of investigations within our class, hopefully. Well, I may have to give you some data, but we will do some data analysis for our laboratory books. So we can share our own data and have a group set. Okay, and when we do that practical, I'll be asking you to think about your variation you observe. Okay, so some of the data will show you that you've got continuous variation, like with our height. So there are, with continuous variation, individuals in that population can be any possible variant between the for example, the smallest and the tallest height. Okay, so which of these do you think will give us continuous variation? Okay, so anything really where you have a number measurement. So height will definitely give us continuous variation. Skull circumference will give us continuous variation, and lung capacity. So this is the size or tidal volume, if you like. Well, not tidal volume, your maximum volume of one breath. But then you will have some that are discontinuous. So whether you can taste PTC is you either can or you can't. It's controlled by one gene. And as we said, colour blindness again is controlled by one gene. So you either are or are not. So individuals for those two will just fall into categories yes or no. Okay, so I'll ask you to do some graphs of this information. So it would be appropriate to use a bar chart for our discontinuous data and to use a line graph for our continuous data. Okay, so you'll maybe have bar charts like this for your PTC, just with two bars, and you might present it like this: percentage of the group. You could just have number of students. But in this case, individuals just fall into one category or the other. This is last year's data for the height investigation. So we did go and add this to a number of years worth of data that I had. Okay, so you can see there we've got most individuals falling at the mean, and if you then move away from the mean in either direction, the number falls until the very extremes where you've got very few individuals at those extremes. This is called a normal distribution curve. Okay, now this is the data that we've collected at John Rigby. Okay, so this if we do the average of the data set, we could work out the average height of individuals aged 17 to 19 at John Rigby Sixth Form College over the last, I think it's about 5 years. If I wanted to compare that to the average height of students at, say, Winstanley College, I'd want to compare the mean values. Okay, but then if I just look at the mean values, it might appear that Winstanley College students are on average much taller than St John Rigby Sixth Form College students. But you've got to be careful, because when we're comparing means, we've got to think about what influence the outliers or the extreme values, so the very, very tall or the very, very short individuals in that population have, what impact they have on that mean. Okay, so when we're comparing variation in two different populations, like between John Rigby Sixth Form students and Winstanley, so we're comparing the heights between these two populations of students, why is it not good practice to just compare the mean heights? Okay, so, well, one, it doesn't give you the variation, doesn't tell you what the shortest student is, doesn't tell you what the tallest student is. And as we said before, if you've got one individual who's really, really tall, it could skew your mean depending how many you've included in your sample. Why is it important to know the variation? Well, we want to know if generally the means are significantly different or not, or whether there's overlap in the population's heights. Okay, so that's why we want to know the variation. So why not use the range? So the range is good, that'll tell us the smallest height to the largest height, and then we could compare those. But what you've got to consider when you compare ranges is that still includes extremes or outliers that are unusual for the population. So as I know we've said previously, what we do use is the standard deviation. Okay, so what is standard deviation? So if you think about that continuous data set, that normal distribution curve, and we put our mean value on, okay, the value that is calculated as the standard deviation is the range, I think it's about 68% deviation from the mean. So it tells us, you know, for 68% of the population of the data, this is the point at which that data deviates from the mean. Okay, so we tend to use double that value, okay, 2 standard deviations, because that includes 95% of the population of the data within that deviation from the mean. And that is a good way of comparing two populations. So we can compare the means as long as we take into account the deviation for that mean for 95% of the data set. Okay, so the other 5% are what we class as the extremes. Okay, so if I take you into your keynotes now, we've tried to show you that with 2 data sets. Okay, so I've given you a definition of range as well. Okay, so why is standard deviation so useful? Well, range indicates the spread of the values from the lowest to the highest. Okay, so in these 2 populations, we've got a spread of 60 for population A and a spread of 8 for population B. Okay, so it is giving us a bit of information about variation, more than the mean. However, the range can be distorted by an extreme value. Okay, so if you look at these 2 populations, the populations appear to be the same in that they've got the same mean and the same range, but the values in the raw data are more clustered in the second population, and the range is being considerably extended by the addition of this smaller value, so this very small extreme, and this very big extreme. Okay, so if we were to consider the standard deviation of those 2 data sets, the deviation from the mean of data set 1 is actually wider, so it's more spread out than population B. Okay, so population B, because standard deviation is less affected by those outliers, when we take standard deviation into consideration, it gives us a better overall idea of variation. Okay, so when you're looking at a graph like my graph here, okay, and or if we look back at this graph here, the normal distribution for our St John Rigby students doesn't look very wide. Okay, so most individuals are falling quite close to the mean value. Okay, so we wouldn't expect the standard deviation to be very large on that data set. Okay, so let's have a look then at using that information. Okay, so as well as being a good measure of variation within a population, standard deviation allows you to compare the means of different populations statistically. Okay, so imagine this is population 1 and that's their mean height. Okay, and this is population 2. This is their mean height. Okay, again, you've got this example in your student notes. So if we bob to that example, okay, so it's actually heights of meerkat populations that we're looking at. Okay, so what does the data in the table above tell you about the height of meerkats in population 1 and population 2? Well, if we only had the mean height data, we would say we believe the meerkats in population 1 to be taller. Okay, now that's a conclusion based on the mean, but then if we start to use our standard deviation, the way we do this is For population 1, you subtract your 2 times standard deviation from the mean to find how much it deviates down, and then you add 28 to the mean to find how much it deviates up from the mean, and that is what you're going to compare, okay, to the other population. So you do the same thing for population 2. Okay, so you take away 91, so that's your bottom value, and you add 91, that's your top value. And if you do that, you can see here that for population 1, we're actually the lowest value. So 95% of the data falls between 144 and 200. Yeah, so population 2, 95% of the range of that data falls between 60 and 242. So a much wider standard deviation. And the important bit is that the top of this one overlaps with this one. Well, they actually overlap a lot, don't they? Okay, so for population 1, it shows that we're 95% sure that the mean lies somewhere between those two values. For population 2, we're 95% sure that the mean lies somewhere between those two values. But unfortunately, the standard deviations overlap, so we cannot say that there is a significant difference between the height of population 1 and population 2, okay, because of that overlap. Okay, so our examiner loves giving you data like this, and you'll see loads of examples in your question packs now and in the future where you have to use the standard deviation. And a heads up, if that column is there, if it is there, they want you to use it, okay? So they want you to do this in your comparison. And then they also want you, once you've done that, to see if the standard deviations overlap or not as part of your conclusion. Okay, so let's have a look at this exam question here. Okay, so the graph shows the variation in length of 86 Atlantic salmon. Okay, what type of variation is shown by the lengths of salmon in group R, and give evidence to support your answer? Okay, if we look at R, it looks like we've got every length of salmon represented all the way from this extreme to this extreme. So we would say that this is a continuous data. We can see it's a normal distribution curve. That's lovely, with the mean higher, and as we move away we're getting lower and lower numbers of salmon in that category. But it's the idea that the data is continuous. There are no gaps between the bars. It's a continuous data set. Okay, I'll show you there. So it's continuous variation. There's a range of values. Okay, no gaps between the bars. You could say many categories, and the idea that they're not discrete categories. So not like with our blood groups, where each category was a different thing. These are not discrete categories. These are just continuous with the next. Confuses some students when they use a bar chart to show continuous data. We're used to seeing it in a line graph. Give 2 possible causes of this variation that result from meiosis. Now pause and have a go. What would we say if it's about meiosis? Okay, so crossing over and independent segregation. It's an old mark scheme, so slightly different term there. But crossing over, so this is exchange of non-sister chromatids of one homologous chromosome pair at the chiasmata, and independent segregation of homologous chromosomes. Okay, so that would be our new spec answer. When comparing variation in size between two groups of organisms, it is often considered more useful to compare standard deviation rather than ranges. So this is like a how science works AO3 question, very common one. So why is standard deviation better than range when you are comparing groups of data or comparing variation data? Okay, so quite a lot that they accepted on the mark scheme then. Range is influenced by a single outlier. So like we tried to show you there in that example in your student notes. Okay, you can say the converse. So standard deviation is not influenced by a single outlier because that single outlier would be in the 5% of the data that's not included within that deviation. So standard deviation shows the dispersal or spread or deviation around the mean. Range shows the highest and lowest values, the extremes, includes the extremes. Now, this next mark point you wouldn't say yet, but by the time you've watched the next video, it'll be something you say quite a lot. Standard deviation is an important thing that is calculated as part of some statistical tests. So standard deviation, we need to do a t-test. So we would be able to go on now, once we've got our standard deviation, to do a full statistical and a test to find out whether or not the difference between the two is significant or not. So this extra bit here is something we'll be able to add a little bit later. Okay, so in keynotes then, sometimes the examiner places something they refer to as an error bar on your graphs they present you. Okay, and they usually tell you that those error bars are 2 times standard deviation. Even if they just say they're standard deviation, you use them in the same way. Okay, so for this population C, the person has plotted the bar showing the mean. So they've calculated the mean and they've plotted that bar to the mean. Okay, then they have calculated the standard deviation and they've plotted the standard deviation down from the mean and up from the mean, so that this bar represents the range, well, not range, the spread around the mean of our data, only including 95% of the data. Okay, so we're 95% sure that the mean lies somewhere between there and there. Okay, now that means when we're making our comparisons, we can say whether or not we think they are valid. Okay, so if you look at B and C without the error bars on the graph, we would say that C has the highest height, so it's got the highest mean height, much higher than B. But with the error bars on, we can say with confidence, because the error bars do not overlap, that this population, the mean height of population C, is significantly higher than the mean height of B, and that's because the standard deviations do not overlap. Okay, so just like when you're using in a table and you're seeing is there overlap between those standard deviations by adding and taking away your standard deviation from your mean, you do the same thing, but they've done the hard work for you. They've plotted that plus your standard deviation and minus your standard deviation on the graph, making your life a little bit easier. Okay, so remember, whenever you see error bars like this on a graph, they want you to use them. Okay, they've not put them there for any other reason than for you to use them. So if there's overlap between the standard deviations in the error bars, you will always say there's no significant difference between these means. Okay, there could be less overlap, but you're still going to say there's no significant difference between those two means. It's only when the error bars do not overlap that you can say wholeheartedly that this mean and this mean are significantly different. So in this case, I'd be able to conclude this one is bigger and this one is smaller. Okay, so just to show you, this was for our population 1 and 2 from the student notes. Okay, no, you won't give me any points for my labels and things on my graph, because I just had to look up which graph it was. So this is our graph to show the average height of meerkats in population 1 and 2. And when I plotted the data, just like when we looked at the raw data set and added and subtracted the standard deviations, it showed the same thing. But it's a lot easier to look at data like this on a graph and say there is overlap, therefore there is not a significant difference in the height of the meerkats in those two populations, compared to, if we go back to, I think I might have whizzed past it, that one, looking at it in raw data like that. Okay, so all I've done is plot that data. So I plotted, this is my bar, and then my standard deviation error bar is plus and minus my 28, and the same for that one. So there is my mean, and then I've plus 28 minus 28. So I'm the same for the second population, but with its standard deviation. And now I can see clearly they're very useful when you put error bars on like this, because quite simply I can jump straight to no significant difference between the means because I've got overlap. Okay, so moving to the end of this topic, this section then, we have got this part in your student notes you can refer back to about probability levels. Okay, so in biology, for our statistical tests, we work at a probability level that the exam board states is p equals 0.05. Okay, so when working with standard deviation, that's using 2 standard deviations to plot your error bars. Okay, remember that includes 95% of the deviation around the mean. We use that probability level for all our statistical testing in biology. It's the same for geography, the same for psychology as well. So if the error bars don't overlap, yeah, and we're working, remember, at a probability level of 0.05, it means then that there's less than a 5% probability the difference between these two means is due to chance. Yeah, so what we mean is it's a really small probability that the difference isn't a real biological difference. So we reject the null hypothesis, and we're going to explore null hypotheses in the next video. Or more likely, what we would say is that the difference between the two means is significant. Okay, so if the examiner tells you it's 2 standard deviations, you can add to your significance. So if there's no overlap between your error bars, you can say that the difference between the two means is significant at a probability level of 0.05. Okay. Right, so we're going to finish there, and we're going to be moving on in the next video to our t-test. So in the meantime, I've asked you to complete a recap on the variation in our variation topic from this one, which I've sent to you as variation summary. Okay, so if you could complete that before the next video, and then we'll crack on with the t-test.